Alright, so I hope you guys have had a wonderful break. I'm not in class today. I'm back in the hospital with Kieran. We had some repeat symptoms, and so we're back in the hospital trying to get those under control. So I'll definitely be out all of this week. That means, unfortunately, I'm not going to see you before your SOL. I know that you're all going to do well. I have no qualms or reservations about that. I know you're going to do well. There is a little bit of content that we still need to talk a little bit about, and that's what this video is about. It's going to be a little bit lengthy. I'm going to try to chunk it for you into pieces to help you get through this content. But it builds upon all of your other understanding of DNA and genetics and Punnett squares and chromosomes and all of that. And it's really about genetic technologies. It's what genetic technologies we have, what genetic engineering is, how we use that. But it is a little out of date. So the technology you need to know for your SOL is a little bit old technology. When I return back to class, we'll talk a little bit more, as long as you have questions about this, and I think you probably will, about some of the new genetic technologies like the CRISPR-Cas9 gene editing technology. So anyways, let's go ahead and get started here and talk a little bit about some of these genetic technologies. So before we do that, I want to do a little bit of a review, make sure that you understand this terminology, because some of this stuff is going to come back, and some of these terms are going to come back as we talk about the technologies. So here's a single chromosome. We don't know from this diagram whether it's an autosomal or a sex chromosome. It's just a model of one single chromosome. We have two sides that are sister chromatids. They're identical to each other. They're connected by a centromere, and together it's a chromosome. And we might even say that it's I'm going to try to label it here, a replicated, just do REP with the dot, replicated chromosome. So these are two sides of the same chromosome. Each side was copied during DNA replication during the S phase of interphase of the cell cycle, using semi-conservative replication. And here's a little model of how genes might be located on a chromosome. If you were to unspin that chromosome or unwind that wound up chromosome, we could look at these sequences, these A's, T's, C's, and G's on this double helix with these hydrogen bonds in the middle. And a section of them, it's probably much longer than just these few, maybe 40, 30 nucleotides. It's probably hundreds or thousands long. But one section might be a gene, another section might be another gene, and there actually might be a section in here that we understand does actually not have much coding significance, right? It just continues to get inherited down from one generation to the next because they're following along on the same chromosome. Sometimes these genes have sections within them that aren't read for that particular protein, and it kind of becomes a little bit more complicated, well beyond what you need to know at this point. But we know a lot more than what you are expected to know. All right, so let's talk a little bit about chromosomes and genes. Here we've got homologous chromosomes, right? So these two right here are sister chromatids. These two are sister chromatids. But they're homologous chromosomes to each other. So the same genes that we'd find over here, like gene A, B, C, D, E, F, whatever we name them all the way down, we'd find the exact same genes on this side for the maternal chromosome, or the chromosome that came from this person's mother, but they might have different alleles or versions for each of those genes. Okay? So when we talk about human chromosomes, key here to understand is we're talking about human chromosomes. We have 46 chromosomes. They're in pairs, so we have 23 pairs, so we have 23 homologous chromosomes. And again, that means 23 of them, one from each pair, is a paternal chromosome that you got from your father, and the other one is a maternal chromosome that you received from your mother. Okay, so the first 22 pairs are called autosomes. This is something that we've talked about, but I'm not sure all of you have kind of clued in and connected in your memory yet. The first 22 pairs are called autosomes. The last pair, what we call the last pair, or the pair number 23, are called your sex chromosomes because they determine your biological sex. The letters X and Y are used to represent those sex chromosomes. They don't look like they don't have the shapes of X and Y. We just called them chromosomes X and Y. Okay? Those are just the names of them. So if your sex chromosomes are XX, then you are a female. You need to commit that to memory. And what that really means is that one of these came from mom, and the other one came from dad. Okay? But, so because mom is a female, she has XX, all she can give you is one of the two X's. That's not true for dad, because dad is, as a male, XY. So if you're XY, then you are a male, and dad can give you X or Y, right? So, because dad is XY, and mom doesn't have a Y at all, that means that this Y absolutely had to have come from dad. This is an arrow. It's not very clear. And then this X had to have come from mom, because the Y is what this individual received from dad. Okay? And you should review your sex linkage genetics here with color blindness and hemophilia. All right? So karyotypes, moving on from these chromosome structures. A karyotype is a chart of chromosomes. They're numbered and matched up with their homologous pairs. We've looked at them. We haven't really studied them all that much. And sometimes doctors will want to see those chromosomes of an unborn baby. They can determine the sex, the biological sex of the child this way. And more importantly, the karyotypes can be used to see if the child might have any birth defects, such as Down syndrome. And sometimes doctors do this kind of screening prenatally or before birth to help prepare parents with one of two decisions, depending on the timing and in what state this is happening. So parents might choose to abort their child, or with this information, better prepare for life with a child with a chromosomal abnormality. And there are many types. Down syndrome is the one that you'll probably see asked about on the SOL, but there are many others that this kind of testing or this kind of chart can help analyze for. So this is a colored diagram. I want to make sure that we understand what we're looking at here. We've got homologous chromosomes paired up, so 2 number 1s, 2 number 2s. So let's see if I can get... Alright, so let's see if we can get... Okay, so 1, 2... All these that I'm... all the ones that are circling in green here, I'll label these in a second once I get them. These are all in green autosomal chromosomes. Autosomal chromosomes. Okay? But the last pair here, I guess I'll do this in yellow. No, these in orange. This pair right here, this is one pair. These are sex chromosomes. Alright? Sex chromosomes. Do my best here. I'm just using my trackpad on this. Sex chromosomes. So, if you count these up, there are 2 for each pair. So this person has a normal diploid number of 46, right? Has 46 chromosomes, and is a male. And so we could write XY, right? And that describes this person's chromosomal karyotype. Oh, sorry about that squeaking. Okay. So because all chromosomes have pairs, this means humans have how many pairs of chromosomes? But all but one of the pairs of chromosomes are ordinary chromosomes. The last pair are those sex chromosomes, or the XY pair. So this is autosomal pair, where these are the same, so the exact same genes on each chromosome, but perhaps different alleles. But the XY is unique because in males the Y is shorter, and therefore there's a section in here that's missing some of the genes that are on the X chromosome, and that's bad news sometimes for males, depending on the condition, because recessive alleles will not be able to hide, like, for example, in color blindness and hemophilia. All right. So again, here's an XY, normal male karyotype, pairs of homologous chromosomes. In this case, they have their sister chromatids with them. And then we've got an XY pair here for the normal male karyotype. Normal female would have 2 X chromosomes for the sex chromosomes. So this is what's kind of interesting, what the karyotype can tell us. It can tell us the gender. We look at those sex chromosomes. If we've got 2 X chromosomes or an X and a shorter Y chromosome, then we know it's a male. But if there are also different chromosomal mutations, and that's key. I want you to make sure you note this: chromosomal mutations, as opposed to what we call point mutations, which are those substitutions that we talked about, like with sickle cell anemia. Like, so basically, it's a substitution. So one DNA nucleotide, like an ATC or G, is substituted for another ATC or G, or there's an insertion or deletion, which we call a frame shift mutation. Please review your notes on that. So this is a point mutation. And these are all of the different conditions of these chromosomal mutations. Somebody could have extra chromosomes or missing chromosomes. They could have partial chromosomes, so some they didn't really travel together, or translocated chromosomes. So trans means across, and located means their position, right? So they moved across from one position to another. Really, this is really kind of going to be the key for our analysis right now and for your understanding. But this is a type of chromosomal mutation. So one of the most common ones is trisomy 21. Trisomy 21. So tri meaning 3, and this is chromosome number 21. And this is critical. I'm going to start next to it. I want you to make sure you jot this in your notes. This is caused when the homologous chromosomes don't separate during meiosis. So look over your meiosis 1 and meiosis 2 notes. In meiosis 1, the homologous chromosomes separate so that you end up with haploid cells at the end of meiosis 1. Well, in this case, what we're talking about is that those chromosomes don't separate. And because of that, you end up with cells that have an extra chromosome and another cell that has a missing chromosome. So let's say the homologous pairs in a woman don't separate, and it's not all homologous chromosomes, but like in this case, chromosome pair number 21, they don't separate. You end up with one cell that has an extra chromosome 21 and the other cell that is missing chromosome 21 because they don't separate. So the term that we want to make sure I want to make sure you know is called non-disjunction. Okay? Non-disjunction. And in non-disjunction, if we want to break that word down a little bit, what we're going to do is, okay, so you know junction means coming together. Disjunction is where they come apart, and non means not coming apart. So these homologous chromosomes that come together don't separate, ending up with this extra 21st chromosomes. So when the one chromosome 21 in the sperm is combined with 2 chromosome number 21s in the egg, because those in the egg didn't separate, this makes a zygote with 3 number 21 chromosomes. And when a doctor were to look at someone's karyotype and see trisomy 21 or 3 chromosome number 21s, they would diagnose that patient or that individual with Down syndrome. So here's a diagram of it. So we've got our 22 numbers here, and then we got XX here. So we know that this is a female, right? A female. But look here, we've got 3 chromosome 21s that are there, and that's what someone would be looking at to diagnose that. So this is not a quote-unquote normal female. This is a female with Down syndrome. Another important one to be familiar with is called Klinefelters Syndrome. And this is when there are 2 X chromosomes and a Y chromosome. So the two X's from mom didn't separate, or the X and the Y in dad didn't separate. And before someone says, well, can you have an XYY karyotype, I need you to think more deeply about why that's not possible. Figure that one out. If you can't figure that out, talk to your neighbors or a substitute, or any other teacher. I think you guys could figure that out with what you know. But here's a very interesting thing. It's been suggested that President Washington, may have had Klinefelters Syndrome. He meets these symptoms, but again, that's matching symptoms. That's not necessarily. There are other things that could cause those symptoms as well. But not being able to reproduce is an effect of Klinefelters Syndrome and having those extra sex chromosomes. All right, and here's another here's a karyotype of that. So you'd see XX and Y for that individual sex chromosomes. All right, so I'm going to switch gears and talk to you about switch gears from karyotypes, because karyotypes are their own kind of field. And before we talk about genetic engineering, I want to talk to you a little bit about cloning, which in effect is a type of engineering, but it's not technically going in and modifying any individual code. There's a procedure involved, and we're going to use some of these terms like diploids and haploids. And so you need to take a pause and make sure you have a clear understanding what diploid and haploid mean before moving forward, because you're going to get a little bit more confused if you don't have this understanding. So I pulled this infographic from the NIH. Excuse me. I'm going to walk you through this diagram of how scientists can clone organisms. Now, of course, bacteria pretty much cloned through binary fission, but sexually reproducing organisms can also be cloned. And we can do this in a like your cells kind of clone themselves when they go through mitosis and cytokinesis. But what we're talking about with this type of cloning is making a new individual altogether that's genetically identical. So we have an adult here that's being cloned. So this orange pig here is going to serve as the individual that we're going to make a copy of. And then we have an adult female who is not cloned and is not actually contributing any genetic material. So what happens here is that there are two extractions that happen. From the pig that's being cloned, they can take skin cells from the adult, or really any other, let me choose a better color here, any other diploid, a diploid cell, so it's got both pairs of each of its homologous sets for mom and dad. It's a diploid. We take those skin cells or udder cells or eye cells or anything else, and then from the adult female that's not contributing genetic material, you take an unfertilized egg out of her ovary, and these are haploid cells, right? So in order for these egg cells to move forward, it's waiting for fertilization, and at fertilization, naturally that's what it's waiting for. At fertilization, the haploid egg and a haploid sperm combine and create a diploid zygote. But what they do in the lab instead is that they remove this haploid egg nucleus, they remove those chromosomes, and then they fuse together these diploid cells and their genetic material inside of them with the casing of the egg. So the egg has all the chemicals and the hormones that are necessary for quick mitotic division immediately after fertilization. So, all right. So the electric pulse will then fuse those cells, and then we end up with a diploid cell. But all of that genetic material is now coming from this orange-colored pig here, and then it goes through mitosis and cytokinesis, and continues to divide until we get this larger mass of cells, which was like a zygote and what we call maybe a blastocyst. It's on its way to becoming an embryo and a fetus, and what they do is they actually implant those cells, that ball of cells, into the uterus of a host or a surrogate, and then after development, after gestation, a pig is born that's an exact genetic match to this adult. Genetically the same, obviously later development, and there are certain health issues tied to that, and there are lots of ethics around cloning, about the value of it, should we be doing it, and people have different perspectives on this. Sometimes it's a religious objection to playing with life, making life. Sometimes people draw that line in different places based on, are we cloning entire organisms or can we clone different organs? And so it might be useful if you had your own cloned organs. In case you need transplant, you could have that exact genetic match as well. We're not there yet, but that's essentially the gist of cloning. And this has successfully been done in lots of different animals, but it is illegal in the United States to do this on humans. Okay. All right, so I'm going to continue on here, but you may pause the video. All right, so Human Genome Project. So this is something I want to talk to you a little bit about. And walk you through how this was done. Alright, so this is the timeline of the Human Genome Project. And I'm going to zoom in on parts of this because there are parts that are familiar to you. I just want to give you the context of when and what happened. But the Human Genome Project was something that started in the 1990s by the National Institute of Health, and it was a 10-year program, just over 10 years, to try to map the location of each gene in the human genome. We'll define what genome means in a few minutes, but I want to give you this context, and so there's a link in this file. If I can get away from, yeah. So we'll walk through this whole, not all of it, piece by piece, but just some of the things that you might find familiar. So, again, we're talking about these chromosomes. You unwind that DNA from around the proteins, and you end up with this double helix. We've got Mendel accredited with discovering some of the laws of genetics, like dominance and segregation, in the 1860s. I think I mentioned this to you before, that his work had been forgotten about. It was rediscovered in the early 1900s. And some work was done, and there's some more, which led later to some development of technologies to discover the structure of DNA, which was done in 1953, credited to Watson and Crick. Remember, Rosalind Franklin was not originally given credit for that. And some more work continued and continued and continued. So we are going to focus a little bit in the technology section on a diagram like this. This is called Sanger sequencing. We're not going to talk specifically about Sanger sequencing for your SOL, but it is a really kind of interesting and kind of sophisticated and concise way to determine an individual's A, T, G, and C sequence in a gene. In the 1982, GenBank was established, which is a repository of gene sequences where you could go and look at that, and we could study that in a little bit more detail if you'd like to. In 1983, the first human disease gene was mapped on chromosome number 4, which is Huntington's disease. And PCR, which we'll talk about, was invented in 1985, which led the way to some genetic sequencing of in forensics that the police used, paternity cases, all because of this 1985 discovery. And then in 1989, cystic fibrosis gene was identified on chromosome 7, and then in 1990, the scientist who was working on the cystic fibrosis gene location was put in charge of the Human Genome Project from the NIH that continued until 2003. There were multiple drafts that were published. I remember when the draft was published when I was in my high school IB biology class, which kind of dates me a little bit, but it doesn't really feel like it was all that long ago. So there was some groundbreaking work done, and so what we're going to do is kind of make sure we understand the big picture of what was done in this Human Genome Project. So we could identify where each gene is located on each chromosome. And I'll show you some diagrams of that. All right, so now we're going to transition to genetic engineering, which we're defining as making changes in the genome of organisms. So actually changing the sequences of genetics. And so we need to define this word genome, like the Human Genome Project, and really it's the DNA sequence of an organism. I showed you a diagram here of a whole host of G's, A's, T's, and C's. And that, again, you should know that's the nucleotide sequence. That's only one side of the DNA strand, because only one side gets transcribed and then translated into an amino acid sequence or a protein. But we could infer and deduce what the other side of the sequence is based on our base pair rule. So G goes to C, A goes to T, and we can complete our model of DNA based on this. But this is a long sequence of nucleotides. That long sequence of nucleotides we can summarize and talk about in terms of dominant and recessive letters, and use capital T's and lowercase t's or whatever letter you want, instead of having to write the sequence every time. So, sorry about that. Bear with me just a second here. I want to make sure I don't lose any text here. So how do we change someone's or something's genome? There are two major ways that we're going to talk about: selective breeding or changing the DNA. And selective breeding was actually something that Darwin uses evidence for evolution as well. And really what it is when we select some traits by allowing only those organisms with the desirable traits to reproduce. And this is not in humans. When it's in humans, we call it social Darwinism or eugenics, and it's not what we're talking about here. We're talking about this in other organisms, whether it be plants or animals, or fungus, or some other non-human organism. So we select those traits, but there are two ways we can do that. It's used in pets, agriculture, meat, or flower industries, but there's one which is a hybridization process, which is where you cross dissimilar organisms to have offspring with the best of both parents' traits. And so, as you can imagine, that may not go as effectively as possible, because all you're doing is kind of randomly mating those that have the best traits, even though they have different types of best traits, and then hoping that you get some offspring that are the ones that you want, that are a mix of the best of both. Excuse me, there's a lot of trial and error involved there. There's also inbreeding, which I think when we think about humans, I think you have a connotation there. But you also, in some ways, we glorify this as well. This is when we cross organisms that have similar characteristics. So think about purebred dogs. When you have organisms that, German Shepherds that have certain traits that you want that are meant to be kept in line with German Shepherds, you mate those that are similar and breed within a small gene pool. Or we can increase variation by inducing mutations. And so this is sometimes done in labs where you have a bacterial sample and you expose them to chemicals and radiation, inducing these mutations. Again, it's kind of random because we can't identify where that genetic mutation is going to happen. It is just going to happen, and if we watch for long enough and watch closely enough, we'll get the mutations that we want. But there's no way of speeding it up. However, now, beyond what you need to know for this, well, there are ways of inserting mutations that we might want. It's just a brand new technology, a couple years old. All right. So we can change DNA by actually cutting it, adding and removing pieces with desired genes. And so we can move genes from one organism to another, for the reason being that all organisms are made out of that same sequence of A's, T's, C's, and G's. But what happens is that we can actually take certain genes that we can use as indicators to know that it effectively works. For example, these are glowfish. You can take a gene out of coral or jellies and insert that sequence into these fish, and they'll glow. You find them at the pet store all the time. They weren't meant for the pet store. They're actually originally thought of as a solution that these fish could be inserted into the environment, into nature, and only glow when there's a certain level of pollutant in the water, and that would be used as an indicator for scientists. But, as you can imagine, when you have a fish that glows, it doesn't stay for very long because they can no longer camouflage. And so they got pulled out of that kind of the line of thinking, and now are sold in the pet trade. We actually depend on that with some of our crops as well. That's how we have tomatoes year-round. They insert a gene that from another organism that slows down its ripening time, so we can get those tomatoes from somewhere else in the world, and they don't spoil before they can get to the grocery store shelf. We can also clone organisms, as we discussed earlier, and so they would have exactly the same DNA. And so we can also extract DNA through a certain chemical and physical processes by breaking up membranes, coagulating DNA. Because DNA has got a negative charge, we can clump it around positively charged structures, and we can cut the DNA. We cut it into pieces, into specific pieces, using these enzymes, and then separating those pieces using gel electrophoresis, and then analyzing that. We can also make lots and lots of copies of DNA. And all of that's required for genetic technology for us to be able to monitor, to identify sequences of DNA. And we're going to talk about those processes. So, about how we cut DNA. So we use these things called restriction enzymes to cut the DNA. They're kind of like scissors that go through, and they cut at specific nucleotide sequences. So here's a little animation. There's this restriction enzyme that is looking for a specific sequence in this strand of DNA, and it's going to cut those pieces. So this is a hydrolyzing enzyme. It goes through it's inducing hydrolysis in the DNA molecule. It splits that molecule, and it's not at the end or the beginning of a gene. It's really whenever that enzyme sees that DNA sequence, it will cut. And so if we're comparing DNA strands, it's not looking for gene for gene. It's looking at the sequence of the DNA, regardless of gene. Okay? They're very, very accurate. We did not do this activity. I've been out, so we haven't done anything with restriction enzymes. But we get these enzymes from bacteria, so we can get a lot of supply of them. So here's another diagram here. So here's an original DNA sequence. We've got this enzyme that's going to come in and recognize this sequence here. Whenever it says CTTAAG, it's going to cleave at this specific site and pull those sections apart. So we end up with a section like this. I would keep going to the left here. We have this middle section that's got 2 sticky ends, and then we've got this other section with 1 sticky end. And so we've got 3 segments here. And then we put them in these gels and separate them. So as you notice that, let me go back here. Some of these can be longer or shorter strands, depending how frequently the enzyme finds the sequence. So some of them can be long, some of them can be short. And so the scientists would mix these DNA fragments and insert them into a gel, right? So it's kind of like Jell-O. And there are these little wells or little holes in the end on one side. I'll show you a diagram in a second. And then a voltage is applied to the gel, where you have electricity running through it. And the DNA is going to move across that gel based on its charge. So the DNA has a negative charge. Remember the phosphates in the sugar phosphate backbone? They carry a negative charge with them. And so they're going to be moving their molecule to the positive end of the gel. Smaller pieces move faster. They can actually move through the pores of the gel faster, so they go further. The bigger pieces are slower. So the bigger they are, the slower they move. And so this DNA that's in these vials are put in with a pipette into these different sections of the gel, and then electricity is turned on. The DNA that's sitting in these wells has a negative charge, so once a charge is applied, it's going to be pulled towards the positive charge, and as the fragments move towards the end, someone turns off the power, and now you have DNA sitting in different sections. So the shorter fragments are farther away from their original starting points, and the longer fragments stay further behind. And then we end up with certain kinds of diagrams that allow us to make some comparisons. Now, if we don't have enough DNA to be able to see their effect on a gel, we have to make more copies of it. And that's done with polymerase chain reactions. So let's break that word down. Polymerase, polymer means many subunits in a large molecule. The ASE is telling you it's an enzyme, and there's a chain reaction. So it happens very quickly. It causes more and more things to be made. So when you have a small sample, the DNA fragment to be copied is put into a solution that's full of adenine, thymine, cytosine, and guanine with their deoxyribose sugar and phosphates attached to them. And the heat will actually separate the DNA into two strands. Remember that there are hydrogen bonds between the two strands of DNA, and that high temperature is going to break that hydrogen bond much more quickly than the covalent bonds along the strand, the sugar phosphate strand. So high temperature breaks those DNA into two, which is the same job of helicase or helicase. The solution cools and the DNA polymerase attaches to the free nucleotides of the open strands. Now you have two identical DNA molecules. You repeat that with DNA polymerase and cooling to make millions of copies of that DNA. Now you might be asking, okay, we don't have helicase here, but we've got DNA polymerase. DNA polymerase is an enzyme that can be denatured with heat. Well, this process wasn't discovered until 1985 when scientists first took enzymes out of bacteria that live in these really hot thermal vents in Yellowstone Park. And by using those enzymes that are adapted to that environment, this heating and cooling did not affect the structure or the denaturing of that enzyme. That's an adaptation that they had for their environment. All right, so they have an original strand of DNA here. It gets heated, they get separated, a new strand gets added, gets heated and separated, and a new strand is added, and it goes over and over and over again. And you can look at the rate here. For each cycle, we've got an exponential rate of growth for the number of strands. So it doesn't take very long to get to millions of copies. So in a crime scene, police collect skin flakes and blood, and they have to ask the question, is there enough DNA in those cells to do DNA testing? Can they identify suspects or victims? And if not, they do these polymerase chain reactions to copy the small pieces. Now, it's not police who do that, it's a lab that would do that with that data. So why is PCR now helping some innocent people found guilty over 20 years ago? Because the technology was not used until the 1980s. So if there wasn't much DNA left behind, police weren't able to identify suspects. But if there were samples of DNA that have been stored properly, they've not degraded, they've not been oxidized, then that sampling can still happen. Okay? And we talked about how come DNA polymerase works at high temperatures? Because it comes from bacteria that lives in these hot springs at temperatures that we could not survive. But they obviously do survive it. Okay? So this is a PCR machine that heats and cools, heats and cools, heats and cools. This is what a gel looks like. There are wells over here that you would insert the DNA into. This is the power box that adds electricity through the gel. Okay. So I'm almost done here, so I'm just going to keep going here. So there's DNA fingerprinting is something that's that you likely will be asked about. It's a technology, so it's like your fingerprints, except it's a little bit more unique than fingerprints. Your fingerprints are not genetically determined If you look at the swirls and whorls on your fingertips, those are developmental. Those occurred during your time in your mother's uterus. But DNA fingerprinting allows us, just like fingerprint analysis of the tips of your fingers, allows us to identify individuals. And this can be used in criminal cases to match suspects or victims to crime scene specimens. Or they can be used in paternity testing. And again, a sample is made, PCR is done to make copies of that DNA. It's added into a gel, electricity is added, the DNA separates out, and then we have an image to compare. So we're going to talk about how we use those in our analysis. So let's talk about criminal cases. I'll give you an example here, and I think if you understand the principles here, then you'll be able to apply them to different scenarios. So let's say that this sample number 1 is crime scene. And we're going to say sample 2 is suspect 1, suspect number 2, and suspect number 3 for each of these samples moving across the gel. If this is our analysis, remember that what we're trying to do is find an exact match. If it's not an exact match, then we're not looking at the same person's DNA. And because every cell in your body has the exact same DNA, because of the S phase and interphase before every cell division, every cell has the exact same DNA, whether it's your blood cells, your cheek cells, your hair follicles, they all have the same DNA. So what we have to do is actually look line by line for each of these sections of DNA that were cut out by the restriction enzymes. And the longer the piece, the further down it is, the shorter it is, the closer it is to the well. And so you have to look piece by piece to find the match. So this is our crime scene column right here. And we notice that this line right here is not exactly lined up. It should start right about here and work its way to the right. So from that alone, suspect 1 is definitely not the person that matches this crime scene data. Suspect number 3 also doesn't match. Sample 3 doesn't match. Sample 4 looks like it's matching. And as I go down and compare to this first column, it looks like an exact match, which means that this person should remain a suspect, while suspects 1 and 2 can, for the most part, largely be ruled out as suspects of the crime. Again, the idea is that you're looking for an exact match for a criminal case. That's not true for paternity testing. So in paternity testing, what we need to remember is that in order to find an individual's father, you also have to rule out the DNA that was provided to that individual by mother. And so when we're looking at this sample here, let me give you some labels and walk you through how we reason through this. So let's say that this sample 2 is the child and sample 1 is the mom. We usually know who mom is because mom was at the hospital and gave birth to the child. The dad was not, the dad's DNA wasn't contributed at the time of birth, but 9 months before that. And so it's sometimes unclear if there could be multiple fathers, which father is the biological father. So if this is the child's DNA, what we need to do is go down the entire row of these bands and determine if that band came from the father. And here's the reasoning that you would use. If the band in the child doesn't match from mom, then it had to have come from dad. It's not a 50-50 split here. We're not talking gene for gene. We're talking about sections of DNA that were cut by restriction enzymes. So if that section doesn't come from mom, that section had to have come from dad. So this first band has this dotted line is coming from mom. The next band in the child is this one, which matches with this sample 3. The next band comes from mom, the next band comes from sample 3, and you can see these dotted lines kind of help us see where each band comes from. And so there isn't a discrepancy here, so number 3 is going to be the father. But if there was even one band that this child has that does not match to dad's DNA fingerprint, then that person, or possible dad's fingerprint, then that person is not the kid's father. Okay? I think that's the end of it. Yeah, so that's the end of it, and that's what you have to know how to interpret and understand. That's going to close out our genetic technologies unit.